Anyways, formally let me welcome our today's guest, Dr. Subhashish Paul. Dr. Subhashish Paul, he did his PhD from the University of North Bengal. He did his master's from the University of North Bengal as well. Uh, his, he did his PhD in cell molecular biology. His topic of the PhD was exploration of anti-inflammatory and anti-arthritic properties of aloe vera from the sub-Himalayan West Bengal. Dr. Subhashish has also been a meritorious student. He qualified UGC JRF Net and uh, on June 2016. He is also the recipient of UGC BS Fellowship for Meritorious Student in Science. Uh, he has also been a college topper during in ABNCL College. He did his graduation from the Kuchbihar ABNCL College, and he has also been the topper over there. He has also received Best Poster Award during the one UGC sponsored state level seminar on animal ecology and behavior. So you can see we have a very talented and very meritorious scholar with us today, and he is going to teach you all today a very interesting topic.
It is
What was
supposed
the
to be
topic?
the immunology of vaccination.
Oh yes, immunology of vaccination. So I personally welcome you all to this today's session, and I especially welcome our speaker, today's speaker, Dr. Subhashish Paul, to our this session. Please, sir, the floor is yours. Please continue.
Thank you, sir. Thank you, Ritesh and Kandikanda.
Just give me a second. I will join you all.
Sure. So, if I start sharing screen, that will be better. But the only problem is that I am attending this particular seminar from my mobile because I am not being able to connect my laptop with the network because it's electrical lack of electricity going on here in my locality. Now, let me start with the screen share and then I'll be talking about the. Okay, am I audible, students?
Yes, sir.
Yes, sir.
Okay. So, the discussion of vaccination came into the story when we all were struggling against COVID, isn't it? So, if you see, like I have written the first page, I have written in the first page or first slide that this is going to be an interactive discussion and not a conventional class. Because if you go through, like as I go through the lecture or discussion, there will be questions raised because we have recently faced COVID. There have been so many discussions regarding the vaccine production process and all, and this leads to so many questions which I will be happy to answer. Maybe a few answers I won't be able to answer you properly, so I'll have to, and you too will have to search it. Okay, however. So the topic of the today's discussion is the immunology of vaccination. If you see, if you have got a basic idea of immune system, can you see the slide?
Yes, sir.
Okay. So if you go through the basic, yeah.
Sorry to interrupt. Can you please increase the screen brightness a bit? Because we are recording the entire thing, it would be easier.
The brightness is that it's high, Shundibanda.
Okay. Then let's go like that. I think we can adjust afterwards.
Yeah, like I can also show you the PPT because the PPT has got some problem because it's not in the laptop.
Yeah, that would be good. Sorry.
Yeah, like if needed, I can provide the students the basic materials of whatever the things I'm going to discuss. If that will be helpful, I'll be sharing those things to you and you can share to them. Okay. So before we start with the vaccination, let's discuss about the basic idea of the immune system. So if we know that immune system is a system which protects our body, you can, if you have got a pen and paper, you can note it down because these are the handy things which will help you to answer the questions in the Viva. So immune system basically protects us in three situations. The first one is when you have got a foreign pathogen or foreign material living or non-living inside your body. The second one is when you have got a altered self cell or altered self tissue or molecule in your body. What does that altered mean? I'll be coming to that. And the third one is if in situations the body produces certain proteins or molecules which efficiently in like blocks the activity of the normal physiology. This is what happens in case of tumor and other cases. But if you look into the immune system, there are four major domains. The first domain is called the innate immunity. You can see it in the slide. That is the non-specific, generalized first line of defense mechanism, something which is given by the skin. So why non-specific and generalized? Because skin is a single protective layer that protects the body against any incoming pathogen. That could be a bacteria, could be a virus, could be something else. And they don't have really a specific mean to protect the body against one of them. So the skin do not adapt a particular mechanism to protect against the virus or rather another mechanism to protect against the bacteria. It's not like that. It's a common general kind of defense. Now the advantage of this defense is this is the primary first degree barrier which will inhibit anything from coming into the body. This is why we need to like put the antiseptic creams and all the other things, or we need to dress. We need to go for the dressing in case where we have got a cut and or injury in the skin. Because whenever the skin is broken or interrupted or hampered, we have got the situation that the pathogens will try to enter the body. The second part or the second most luxury kind of immunity we have got is the adaptive or acquired immunity. This is a specific kind of immunity, often involves genes, the genes related to the immune system. This is the second line of defense against the antigen exposure. It is generally mediated by T cells, B cells, and any of the, to be precise, any of the like WBCs we have got, they have got their specific kind of role. Now, adaptive immunity is specific because the kind of mechanism the body would adapt against one particular virus might not be the mechanism for another kind of virus. So the mechanism should differ, and in the immune cells, the genetic rearrangements leading to the production of a particular type of antibody or maybe a particular type of activity of the cell will be controlled. You will have like the immune cells will have to produce the cytokines, interferons, and so many other things are there. So the production of the proteins and all those things will need the genetic rearrangement or the inactivation or activation of the genes. Now, the immune system, generally the adaptive immune system has got two major domains. The first one being the humoral immunity, where it's more like a sniper. So the immune cells, they don't go and kill the particular pathogen. What they do, rather they sit in somewhere else, they produce certain proteins which goes and interacts with the pathogen and kills it. So it's the antibody secreted from the B cells does this thing. There are so many other molecules in the body like the complements and the small peptides which are like which can break the proteins, the proteases, those are there. So the immune cell do not directly interact with the particular type of pathogen, rather it sits somewhere else, works, uses a sniper and kills the particular pathogen. The fourth one is the hand-on combat. That is the cell-mediated immunity. So the immune cells directly kill the pathogen by eliciting some kind of responses related to enzymes or maybe some other molecules, or they phagocytose the cell and so many other things happen. This is happen which in like One such example is the NK cell killing the tumor cells. So these are the major four domains of the innate immunity, the immune system, the innate one, the adaptive one, the humoral, and the cell-mediated one. Now, the immunization, the story of immunization has been a old story in the field of immunology. So, like in many a times, people have seen that one individual infected with a particular type of disease cannot spread the disease to all. Rather, a few other individuals are infected and the rest are okay. Something which we saw in case of COVID as well, when you see that one particular family member is seriously ill with the COVID and the others are not, some are showing symptoms, some do not. Some healthy people whom would you find like the hatta katta type of people जिनको देख के कभी लगता नहीं है कि they can die out of COVID or something like that, they have died. A person with a very, maybe he's very underweight type, like suffers the influenza and all the other types of symptoms all through the year, but that person survives the COVID. We have seen all the, all the mix-ups in during the period of COVID. Now, the COVID has been a like big role player in the development of recent theories of vaccination and immunization, and so many terminologies were again heard during that particular time. But immunization is basically, it started during the period of Renaissance by Louis Pasteur and Edward Jenner. They did a lot of experiments regarding the immunization without even knowing the kind of immune system we have, because the technologies we used at that time was very primitive type, and the molecular techniques, those were not developed. Still, the immunization has been defined as the introduction of an antigen or an antibody to an individual to boost the immune response of the individual against a specific pathogen, leading to immediate or long time release from the infection. So we have got the vaccines against the polio, diphtheria, Japanese encephalitis, then we have got smallpox and all. And these diseases are almost 100% controlled with the use of this particular immunization. If you go into the process of immunization, there are two major types of immunization. The first one is the passive immunization. It is a situation where the exogenous antibodies from another individual or some or related species is provided to an individual in an attempt to prevent or attenuate the anticipated infection. So this is basically a situation where you collect the antibody from one individual and deliver it to a patient. Now this antibody collection and delivery is commonly known as the serum transfer technology. The serum transfer was discussed during COVID. I will relate most of the topics with COVID because this is something you have faced and you know. So there was a discussion during COVID that a few patients survived when the serum from a like seriously ill or symptomatic patient was delivered to their body. This is because when the particular person's immune system is weak, you are simply supplementing that particular person's immune system with some procured or like isolated antibodies, and these antibodies which are very generally secreted by the B cells against a particular pathogen. In this case, that is the COVID provided. So you are basically sending the army to a particular individual's body just to make sure that whatever like strength that the immunological strength the person is having, that will get increased. But the passive immunization has got a short-lived effect because all the proteins in our body, they have got half-life, so they will eventually break or they will eventually get digested by the protease enzyme. Similar is seen in case of the antibodies. So if you inject a particular person with someone else's antibody, the antibody will be there in the circulation for a period of time and then it will get vanished, it will get digested and you won't see the effect. The passive immunization is also procured from mothers as well. You see, the maternal antibodies transferred transplacentally initiates the natural passive immunization in all the humans. So there are types of antibodies which can pass the placental barrier, and these antibodies provide a parallel defense mechanism in the newborn or the placental body as well. The next part is the active immunization. So this is the part which we are going to discuss, because vaccination is a prime type of active immunization, where what we do, we just stimulate the immune system. We stimulate the immune system to make sure that the immune system reacts in a faster and better way when actually the pathogen comes into the body. Now, there are so many sophisticated domains of Indian Army and Indian Navy and Indian Air Forces. The guerrilla warfare, the kind of tactics we see in case of the Marcos of Indian Navy. They are especially trained, and if you have a bit of interest about these things, you will see that many countries they go for the joint venture or combined workshops regarding the modes of the army interaction. So we have got the army practices between Indian practices between Indian US Navy. In newspaper you will see all these things. And why this happens? Because this is basically a transfer of technology and thought to make sure that if a particular situation comes, what the army or the particular group of the military should do. Now, this is also the prime kind of thought which is seen in case of the active immunization. So, consider our body to have the army, and you put the antigens or the pathogens at a very low amount, so that the army can get trained against that particular antigen and pathogen, and the training will help them to identify the same kind of pathogen if it enters again, and they will show you a higher and faster kind of response. So the active immunization is the process of boosting the immune system of an individual by introducing harmless antigens at a very low dose, which engages immune cells in a way that the cells respond quickly when the pathogen enters the body in actual. This is something which happened during the COVID. Okay, I'll throw you a question if you are listening to me. The question is very simple. You have heard that doctors, they were more prone to COVID, or doctors were dying, or the people who were associated with the health services, they were dying more compared to the normal people during the COVID. Have you heard that, students?
Yes, sir.
Yeah. So, do you have any idea why this happened? Any idea or any thought, like why doctors died more, or why doctors or other health service providers were more susceptible to the particular disease? Is there any relationship between such a thought that the virus entered their body in a in a maximum in an increased manner compared to the normal people? Is there any thought like this?
So, like,
Yeah.
there are more chances.
Okay. See, there is a term which is used in the virology as well as in immunology, that is known as the pathogenic load. You can note it down if you want. So, pathogenic load is basically a situation where, like in our body, if you see, one virus entering your body never assures that you are going to have the symptom. Any virus, you consider it the influenza, you consider it any viruses, a low number of viruses or pathogens entering your body, your immune system is always fighting against that, but your immune system has got a particular limitation. So, like it's more like you cannot control a group of 1,000 hooligans against, like with the help of 10 police. You need to have a bigger kind of army or police or like security guards to control 1,000 hooligans. Now, when the number of hooligans are less, that there is no problem. A low number of viruses entering your body, immune system is killing them or like neutralizing them, so you don't have the disease. But regarding the health practitioners, because they were working with the COVID infected people, and COVID is a virus which like spreads through the aerosols or through the air, like the water the vaporous outputs of the nose or nasal cavity. So if you are like stuck in a congested room with so many people having COVID, so obviously the number of viruses in that particular room will increase, and no mask, nothing can prevent the virus 100%. So if the number of viruses in the room increases, there will be a higher chance that the the more number of viruses will be entering your body. That this is what happened in case of doctors. If more viruses are entering, then obviously the person will have a higher viral load, and this higher viral load is more like having thousands of hooligans against 10 or 15 policemen. So the hooligans would take the control of the situation, rather, or the virus will take the control of the situation of the doctor's particular physiology. This is why the active immunization process is kind of treatment which is given to the people who are more prone to the exposure of higher number or even lower number of the pathogens, and this particular process basically initiates with the introduction of the virus or virus-like particles into the body at a very low dose, which can actually evoke an immune reaction, but not at a systemic level or not at a level where it initiates the disease. And the virus vaccination against COVID was such a situation where we found that people were getting immunized, and even after that, they were having a low dose of the particular disease. But interestingly, a very few number of people died after they got immunized. Now, let me tell you one thing. Post-COVID, in the post-COVID situation, we have seen nutritional supplements and so many other things associated with the immune boosting and all. Now, trust me, the immunity is 95% genetic, and if someone is not having any malfunction, malnutritional issue, the immunity cannot be boosted to a situation where someone who was prone to death will become like immortal after the particular supplement was taken. This never happens. So the genes has to be there. There is the term which was also coined or which was also discussed during the COVID, that is known as the cytokine storm, which controls the internal environment of the immune system of a particular person's body. If the cytokine storm is okay, which is obviously genetically governed, if the cytokine storm is okay, you see the person surviving, and if there is some mishaps with the genetics, then the person will not survive. This is as simple as that. Now, the active immunization, the prime type of active immunization is vaccination. Edward Jenner and Louis Pasteur initiated the specific practices of vaccination. It's a long-lasting, effective immunity against a specific infecting agent with the help of immune-competent cells of the host body who are exposed and trained, as I said, to kill the pathogen. Generally, B cell dependent immunity leading to the clonal selection theory is the main mechanism against that particular vaccination process. So, let me tell you about the clonal selection theory, because this is the theory depending on which the vaccination process is generated. The clonal expansion of a cellular subset of T and B cell against a pathogen. So, whenever a pathogen enters the body, my body or anyone's body, there is a pool of T cells and B cells. So, the T lymphocytes will be sitting around the thymus, the B lymphocytes will be sitting around the bone marrow, and they would get the pathogen. The pathogen will be carried to them, or maybe some cells will get infected and the cells will secrete the cytokines. So from the bone marrow and the thymus, different cells, the different immune cells will start migrating towards the particular place of infection, and they will find that okay, there is a pathogen, and all the T cells and B cells, they will start their clonal expansion, or rather simply if I tell you that they will start their division. And as the division starts, some of the cells will be smarter to identify and react against the particular antigen. So if you see the basic structure of the antibody, The FAB portion of the antibody has got variable protein sequences, so the 3D structure of the antibody differs at the FAB region. So not all the antibodies can bind strongly with all the antigens. There is a specificity where one particular antibody can bind with one particular antigen. Now, the B cells, there are so many B cells in our body, but the type of B cell which has got the capability of producing an antibody which binds more stronger to that particular pathogen which entered your body, that particular B cell is known as a clone of B cell which is more reactive against the particular pathogen. Now, this particular clone of B cell will be selected and activated in the body. So, the body will somehow, using the different molecules and cytokines, will upregulate the expressions and the divisions of that particular B cell. And if this happens, what happens? The differentiation and proliferation of the clones into effectors and memory cells will take place. So it just got a bit mismatched in the PowerPoint. I'll send you the correct one. So there are two terms. The first one is the effector and second one is the memory cells. So effector cells are the cells, the B cells, which will immediately start showing the effects. That means they will be killing the particular pathogen. And the memory cells what they will do? They will not produce the antibody. They will not participate in the pathogen killing. Rather, they will sit in the body and they will go on they will be doing their division and differentiation. So, if the effector cells they kill all the pathogens and the pathogen is cleared from the body, even after that, the memory B cells will be sitting in the body somewhere and they will go on, they will do their divisions and all, making a bigger or a larger pool of such B cells. This happens if the same pathogen enters the body, maybe 6 months or 8 months back, 8 months later, then these particular memory B cells will have the memory to identify the particular pathogen and they will kill the pathogen in a more proper and faster way by changing them into the plasma cells or the effector cells. So this is what happens in case of clonal selection theory. So there are different clones of the B cells and T cells in the body. One particular clone can identify the pathogen in a better manner and that particular clone gets uplifted and their translation, transcription and their cell divisional properties are upregulated and they make a pool of the effector cells and the memory cells. The memory cells are kept for the further exposure and the effector cells they kill the particular pathogen. Now I come to the advantage of the vaccination. So the vaccination has got so many advantages. The first one being, as I said, that it provides a long-term immunity against a particular pathogen. The second one, this term herd immunity and not hard one. The second one, this term herd immunity and not hard one. This herd immunity was also discussed during the COVID. Herd immunity is also called the community immunity. So the other advantage of vaccination is that it can actually encourage the development of the herd immunity in a community. So suppose a virus enters the body, there is always a relationship between the virus and host in such a way that no virus wants to kill the host. This is true. Because what happens if the host is killed, the virus won't be able to infect the same individual again. So there will be a lack of host if the virus starts killing the host. So virus, they always have got a tendency to become a less virulent form where the host will not die, rather the host will get infected, he will get cured, and there will be a second and third and fourth chance that the virus will infect the host. So the host pool or the host population will not get reduced. So if the host population is not getting reduced, obviously virus will survive because virus is something which can survive only inside the host body. Outside the host body, it just looks like a dead kind of thing. So no virus wants to kill a host And this is why the viruses they often shift to a less virulent form. Something which happened during the COVID as well. The primary forms were very strong, and then we had the weaker forms of the or weaker varieties of the viruses, which were not killing the host efficiently. Rather, it was like a symptom of influenza or viral fever. And there was the advantage, there comes the advantage of the virus where it could infect the same individual again and again. Okay. Now the herd immunity is something which is seen when you start the immunization or vaccination process in a population. If someone is immunized or vaccinated, he will have the capability to fight against viruses and he will not show the symptoms. He will not get bound with the issues and symptoms of the disease. So there is a less chance that the person will be spreading the particular virus. So as more actively immunized people are there in the population, the more will be the prevention of the pathogen. This is known as herd immunity. So when India reached about 60% vaccination in its population, people started saying that the herd immunity has been achieved. So most of the people are immunized and the disease will not get spread easily. Okay, so these are the two advantages. Now there are some certain disadvantages. Obviously, you need to take care of the cost benefit thing. So the companies they use billions or trillions of dollars to make or to produce the particular vaccine, and it's a complicated trial and error process. It has got so many steps of clinical trials through which you need to go through. You need to test it in a mammal first, then into the volunteer humans, then you have to market, commercialize it. Also, you have to take care whether the person who has got the particular vaccine has got any other issues or not. So there are so many things. The side effects has been a concern. There has been side effects with AstraZeneca vaccine. People were discussing that the people were dying out of that. So what the paper said is that if the person was dying, it was between the first few months, and it's very abnormal to find a person dying out because of that particular vaccination after 5 to 6 years. So this generally it hasn't been proved. Now coming to the types of vaccines. So in this particular slide, I have got the types of vaccines which are more used. So the live attenuated vaccine is one which is used in most of the cases where the virus is kept or kept live, but their virulence property is deleted. So you need to delete a part of gene maybe or part of the genome maybe, which makes them a virulent form. So they can actually develop and they can actually grow, but they cannot infect the particular host. Something which is seen in case of the BCG vaccine. The other part is the killed vaccine. The most important or the most convenient one is the heat-killed vaccine, which is seen in case of polio. So in polio, the heat-killed vaccine or the heat-killed pathogens are introduced into the body of the particular individual. As they are heat-killed, they do not show the property of growth and becoming infectious. Rather, they are simply killed ones which enters the body. The body identifies the capsule proteins and their structures and develops a particular immune defense mechanism against that. So, in reality, when the virus tries to enter the body, they would again identify the capsule structure and would find that, okay, this is something I have seen already, so I have to show this sort of immunity, and the particular body reacts accordingly. The next one is the toxoid vaccine. The tetanus toxin inactivated. Inactivated tetanus toxin is given to the particular person's body. There is a toxin. There is a toxin which is made by the particular pathogen. That which is actually the particular prime molecule which infects creates the symptoms and disease. So that particular toxin is isolated from the virus body, and you have to develop the particular kind of vaccine. You develop the particular kind of vaccine with that particular toxin. The neutralized or inactivated toxin is called toxoid, and against the toxoid, you find the immunity in a person's body. The other one is the purified antigen vaccine, which is seen in case of typhoid. So you can kill and break the particular pathogen and find a few of its more potent capsule proteins or other or other molecules or sometimes the polysaccharides which are unique to that particular pathogen, and you develop a vaccine using that particular thing. So the purified antigens from a particular organism like Salmonella typhi, if you purify it, the polysaccharides and the proteins, and you inject it in an individual's body, then the individual will react and will make sure that the immunity is developed against that particular purified antigen. The other advanced new generation vaccines includes the recombinant vector vaccines, the nucleic acid vaccines, and the anti-idiotypic antibodies. So recombinant vector vaccines means it's more concerned about the recombinant DNA technology where you take a particular DNA of interest which causes a particular disease and you put it into a vector's body and send the particular vector containing the DNA is given to a particular organism and the particular microorganism is injected to a person's body. These are the nucleic acid vaccines and the idiotypic vaccines. I don't think you need to go through all these things because the processes associated with it are very complicated. However, but in most of the cases, it needs the fundamental and advanced research. The HIV, in case of HIV, the scientists are working on the recombinant vaccines and the anti-idiotypic antibodies, but it hasn't get standardized yet. So the time will be taken because for viruses, they always find an escape route. They always find an escape route to survive because what we do, we kill the viruses and they have to find the escape route to survive, so they get mutated faster. And there is no such DNA repair mechanism which can prevent them from having the mutations. So as they change faster, their code proteins and all the other antigen factors also change faster. And this is why in many cases you cannot develop a vaccine because the vaccine you are developing now might not be efficient against the particular against a new variety of the same virus a few years later. So this lies the problem with the new generation vaccine. Okay, this much for today, I guess, because I have given you maybe a cluster of information which you are not aware of and you need to study a bit. So I would like to ask you if there is any question.
স্যার আমার একটা কোয়েশ্চেন ছিল স্যার আপনি ভাইরাসের একটা ক্যারেক্টারিস্টিক্স বলছিলেন যেখানে আপনি বললেন যে ভাইরাস প্রবাবলি হোস্টকে কিল করতে চায় না, কিন্তু স্যার ভাইরাস তো সবসময় হোস্ট সেলে গিয়ে ওর ডিএনএটাকে ড্যামেজ করে এবং নিজের ডিএনএ ক্রিয়েট করে তারপরে সেল লাইসিস করে সে বেরিয়ে আসে।
না, এটা কি ব্যাপারটা দেখো যে ভাইরাস হোস্টকে কিল করলে যে ভাইরাস হোস্টকে কিল করে, ধরো এরকম একটা ভাইরাস যার কিলিং প্রপার্টি অনেক হাই, সেই ভাইরাস যদি আমাকে তোমাকে ইনফেক্ট করে এবং আমরা মরে যাই তাহলে নেক্সট টাইম ও কোনোদিনও পাবে না। অ্যাকচুয়ালি ভাইরাসের তো আর এই লজিক নেই যে ও কি করবে, বাট বেসিক্যালি বিকজ দে কিল more and more, so the particular variety cannot get spread, এবং সেই জন্য যেগুলো হচ্ছে virulent forms, যেগুলো মানে কিলিং রেট অনেক বেশি, সেই ভাইরাসগুলো জেনারেলি পপুলেশনে খুব বেশি দিন স্প্রেড করতে পারে না, because whoever they are infecting are dying. Now what happened in case of COVID? There were so many asymptomatic people যাদের মধ্যে ডিজিজের সিম্পটম দেখা যায় নাই, they were moving around এবং they were spreading the disease. How about a situation where COVID kills the person, most of the person within maybe 3 to 4 days? তাহলে কি হতো, যাকে ও ইনফেক্ট করছে সে বেড রিডেন হয়ে যেত, সে মুভ অ্যারাউন্ড করতো না, তার থেকে ডিজিজটা স্প্রেড কম হতো। So the virus with better killing property has got a less spread, এবং এই ভাইরাসগুলো সহজেই পপুলেশন থেকে মানে হোস্ট বডি থেকে এলিমিনেটেড হয়ে যায়, because the host they do not live. Only the varieties of viruses which survive is basically a situation where virus can infect you, you will feel sick, but you won't die. So the viruses with a less virulence has got the tendency to spread over the population for a longer period of time. আমি এটা বলছিলাম, এবার বুঝলা?
Yes, sir.
Okay. Any other question in this regard?
Any other questions from any other student? Yeah, I must hope.
Any other questions from any other student?
Because
I must think that you may have so many questions because it's such a fantastic topic and something which we have all experienced over the last 2 or 3 years, and there are so many stigma, so many common people's knowledge that is connected to this COVID and vaccination. I think this is a grand opportunity for you to test what you think and what you believe and what actually is. Go on, student.
Good evening, sir. Sir, like जो natural
Good evening,
selection
sir.
से viruses and bacteria वो vaccines को
सर
adapt नहीं कर
लाइक
सकते।
जो नेचुरल सिलेक्शन से वायरसेस एंड बैक्टीरिया वो वैक्सीन्स को अडॉप्ट नहीं कर सकते।
it's not a part of.
Yeah, there is
it's
a
not a
chance.
play of one or two.
But still it takes time. It's not a part of, like it's not a play of 1 or 2 generations.
And after
So, sometimes it happens that a particular vaccine is developed and after a few generations maybe the particular vaccine is not working or the functionality or effectivity of the vaccine changes between generations. This happens. But like vaccine development is a different kind of thing, so you won't see any vaccine being less efficient because the labs, they are also developing newer techniques to generate the second and third generation vaccine. But if there is a chance that the virus it's not about the virus, rather I would say that it's not about the virus of getting adapted, but rather, as I said, that viruses have got a chance to mutate faster, to get the mutation at a faster or higher rate. So generally, a new variety of virus would come, which will show a different set of proteins, which will be not detected by the previous kind of vaccine. This happens. This is something which is very common. Even if it happened during the COVID as well.
Okay, good question, Garima. Anyone else?
Okay.
Okay, good question, Garima. Anyone else?
Topic is directly related to your syllabus.
Other students from 5th semester, because this topic is directly related to your syllabus.
Sir, I have a question regarding this. Similar type of question actually. Sir, जो आपने बोला कि मतलब जो ये वायरसेस है जो म्यूटेट करते रहते हैं, तो एक बार जो अगर एक वायरस ने हम लोगों को इन्वेट किया है और हम लोगों को एफेक्ट करके हम लोग जब वैक्सीनेट हम लोगों को हो गए, इम्यून हो गए हम लोग, लेटर ऑन वायरस फिर से म्यूटेट होगा तो हम लोग इम्यून होंगे या फिर उससे फिर से हमें वो हार्म हो सकता है?
It's a very good question. Like, let me tell you, बहुत बार हम लोगों को इन्फ्लुएंजा होता है ना, वायरल फीवर.
Yes, sir.
This is because once you are infected with a particular type of strain of the influenza virus, you won't get the disease again. That is true. But maybe next time, in a gap of maybe 2 months or 15 days, if you have got influenza again, then obviously there are the sets of memory B cells and other cells in the body. But that particular second type is a different form of virus, a different variety of virus. So there is a chance that if the virus has the havoc mutation and changes a lot of proteins, so the virus can infect you again and again. It's the different variety. This happens. Influenza में वही होता है. Like we have got around maybe Shondipanta or Rideshda knows it better. Have got around 3,000 to 3,400 varieties of influenza we have got, and each variety can infect us. So even if you are immunized with one particular form, this never assures you that you won't get infected from another form. This happens. This is true. But this question is very good, and it happens in case of the influenza.
Just to add a
Thank you, sir.
Yeah, just to add a story here. I mean, I had done one,
article on this.
what should I say, popular article on this discovery of virus. And there actually I was going through many of the, previous historical documents which were done at the time of discovery of viruses. And initially, for 50 to 60 years, people were very much confused whether it's a virus or it's a very small bacteria, because only in 1950s, around 1930s, people started to suspect the existence of the virus, and then from 1950 onwards, people were sure that a virus is such a thing. Because before that, before tobacco mosaic virus being, imaged by the process of electron microscope and the XRD, people had no idea that such thing ever existed. But to add to Dia's answer, that possibly this is the biggest dilemma in the entire, science of virology and infection biology, that it also gives us a drive that we must not stop, because this is a course of research, this is a stream of research which must go on. And here we need, possibly some of the students from this group may start a new, journey in the future also, because this is a stream of science which will never be blocked, because there is a continuous tug of war going on between us and the viruses, and nobody is the winner. Sometimes they put us two goals, and now we have put two goals to COVID. Now COVID has gone. Yeah, we lost. All of us have, severe casualties, both in terms of psychological, personal, financial, economical, and yet again we are alive. We are again roaming, but the COVID has been, managed. This is the thing, if you study in history in 1918 and 1920, there was influenza virus. That's possibly the biggest pandemic, the, greatest pandemic in
It was in,
humanity.
sorry to interrupt, it was in 1910 probably when we got plague.
Yeah, plague. The only plague was, and then after
67% populations of India that time.
yeah,
In the Bay of Bengal, it was more evident. So, now, if you see statistically, Sundibanda, the fun part is COVID has killed less than 1% of people. It's about 0.4%. Now, if you consider 0.4%, death is a very havoc, destructive kind of virus. What if some new varieties come with a death rate of 10% or 15%?
But there are many political issues too.
So,
yes, I agree, but there are many political issues too, because in plague at that time of India, our colonial rulers never gave us the true figures. The number of deaths of Indian people, the natives, when none of them, possibly only 2 to 3% of them was, calculated, because it was a big gap on their medical facility also.
More than that, way more than that. Way more than 5%. Because in India, in 1910, the population was around 30 crores. So it has must have been a more number of deaths compared to the official documentation. But even in today's story, that we have got a very sophisticated lab. I don't know the present situation though. Because must be knowing. Shandipanda also knows. NICET, we have got NICET in Kolkata, National Institute of Cholera and Infectious Diseases. They are working against the infectious diseases and viruses, and they're trying to develop the pathogenesis and the medicines and whatever. Now they have got the IICB and TIF are working. So there are so many labs where the students can get the exposure. Now, discussing about the viruses, discussing about the vaccines and making a vaccine, they are way too different, because you have to look into so many small and big things, like when you develop a particular vaccine, because it's always a question of life and death. And as we have developed a few, India has developed a few, the world has developed a few vaccines at a very rapid pace during the COVID. So this has got some interest, that how the vaccines are developed and what are the techniques being used. And this is a new domain of research that students can actually look into. So there is so many things to do.
I don't think there are any more questions from any more students.
I don't think there are any more questions from any more students. So let us formally I think I can see a hand. Yeah, Ruhi possibly. Yeah, go on, Ruhi.
Good evening, sir.
Yeah, good evening.
Sir, when the epidemic was going on,
There were like many cases of COVID.
and there were like many cases of COVID. In the news channel also, it was like horrific, listening to all the news, the number or the population of people dying.
Yeah.
There was not a single case, or maybe not a single news related or that said that a person, a beggar or a person, street person died.
And
It was
there
not.
are, yeah,
It was
I
not
can
heard,
get you.
sir.
Yeah, see, there could be two answers. I don't know if both are correct, or maybe it's my personal opinion actually. See, during the COVID, no one actually took care who died. So even if they have died, if someone staying on around the streets or somewhere, if they have died, and if the municipality has taken the body to somewhere, you won't be knowing. Maybe that's one thing. The other part is, the vaccination process, or the not the vaccination, the immunity depends more on exposure. That's a very fun fact. So the more unhygienic life you have, the more is the antigen exposure, the more stronger immunity the person should have or will have. This is the unsaid truth. You bring someone from a very sophisticated, hygienic country, the first world countries, you put them somewhere near NJP station or maybe somewhere near Kolkata or Delhi, they will definitely suffer a lot of problems because they don't have the trained pool of T cells and T cells in their body because they are not exposed against those viruses or pathogens. Now, when we have a kind of semi-hygienic life, or I would try to tell it an unhygienic life, we're exposed to so many pathogens, the street foods, whatever we have, the level of pollution we are having, these are the issues where, these are the situations where the immune system actually gets boosted. Now, this boosted immune system gives you an advantage over a bigger array of antigens to fight. And this is why maybe the people, you are asking about COVID. I would ask you the same question. See, have you ever seen a person the stray person and the vicaries and all? Have you ever seen them suffering from fever? Have you ever seen them suffering from some other issues which are visible? They don't. Because what I feel is that they have got a bigger pool of activated immune cells, and this is what provides, this is what protects them. Now, I don't have any data. Maybe nothing was done. No record has been done regarding these people. But the two, both the reasons could be right. That one, that documentation was never done because they died and maybe the body was thrown away or kept taken away by the municipality or other authorities. This is the first one. And the second part is they have got an increased level of immunity. This will happen because they have got the unhygienic, sometimes rotten food. They have got the unhygienic kind of living. They don't have a room to live. So their immune cells are more boosted. Their total immunity is boosted. So there could be this could be another reason of survival. I don't know if Aritesda and Sundibanda can add anything else to it. This is probably the only. These are the probable reasons where you can look into.
I think he's very much. I think he's very much true to what he said. People. That's a. I mean that's a unsaid truth that people who are exposed to more pathogens, they tend to develop more immunity. It's a very simple formula. Once you, that's why people who are more into street food, they don't get usually stomach upsets. The people normally who take very healthy foods, hygienic foods, whenever they go outside and take any street food, they tend to have stomach upsets. So that's very common. Exposure gives you more immunity. Okay, and that may be one reason, and the other reason may be that the people who died, people were more concerned about the usually high-profile people or people from the middle class or well-known people. Okay?
Yeah, they are basically out of the society, so no one thought about it.
Basically, when you are at the bottom of the strata, societal strata, so nobody bothers about your death or your life. So that may be another reason. So these two may be the reasons why you didn't really find those kind of data.
Okay.
Yeah,
Do you have got something else to add?
Yes, sir. Sir, suppose a normal person is exposed to a pathogenic environment or something like an unhygienic environment. So, how long will it take for the body to, develop antigens against it?
See, the question, it's a question which has got a very generalized kind of idea. So you are talking about the pathogenic environment. So how many and what types of pathogens are there is the first question. The second one is generally body, like the immune system has got so many cell types. each one each of the types are working at their own way, but ultimately providing a basic goal of ultimately providing a basic protection to the body. So you know there cannot be a question like exposed to a pathogenic environment. The question must be like how many pathogens are there. If you talk about science, that how many pathogens are there and how much is the virulence, what kind of pathogen is that, whether it's a bacteria or a virus. So all these questions, all these scientific things come into the story. But it's more about upbringing someone. If a child is exposed to the outer environment, if he plays, if he goes around, if he gets cuts and the injuries, those are the normal things which maybe your generation is not facing. So you people don't go to the fields, you don't have got cuts and injuries and all those things. So the pathogen you consider to be a pathogen might not be a pathogen for us. Me, Riteshda, we have been playing cricket since long, Riteshda. You can remember the old days. So we have the cuts and all those things. So there is nothing like pathogenic environment. You have to be like accurate regarding what pathogen you are talking about. If you are exposed, like if you are around the dumping ground, and the dumping ground is about the household thing, then there is a different set of pathogens. If you are roaming around the hospital and you see the like dumpings of the hospital effluent, this is the second set of pathogen. Now the kind of immunity you develop for the first set of pathogen and the time it takes might differ from the for the second set of pathogen. There lies the problem.
Okay, I think you have understood the reply from Subhashish. I think there is no more questions today. So thank you, Subhashish, for such a
Thank you very
fantastic talk.
much.
Thank you. Yeah, we
Wholeheartedly
met.
thank you and Riteshda for this particular opportunity.
Yeah, I think our students must have enjoyed it, and it's a fascinating topic. It's a fascinating field of research and study. I mean, immunology is something which I personally always enjoy, and all of us possibly, because we come from we do hold a legacy of this particular topic in NBU, and I have the privilege to represent many other universities for my professional research career, during my professional research career, and wherever I went, luckily or unluckily, I have been always tagged as a immunology specialist only because of the training we used to get in NBU. And I must also share to our students that possibly this particular topic is being taught the best way, at least in West Bengal in NBU, because it's something which we all take pride in, and I think possibly in near future we may find some of our students also. They are also taking part in the continuous progress and the development that we have been made in this particular field. And on this note, I would request HOD Department of Zoology, Dr. Ritesh Bishwa, to formally conclude this program and provide a vote of thanks.
Thank you, Sandeepan. And I have been throughout this entire session. I was just looking at how how Subhashish was presenting the whole presentation, and the way he presented the whole concept considering the COVID. It was really mesmerizing, and I kind of felt proud in the sense that
It's all because of the trainings I got during my PhD, and you people were
absolutely,
here. it's a legacy being carried forward, Ritesh da.
Absolutely, and that's what we had a history. We have a history of connected during his university time as a student and also as a scholar, and we worked together. And looking at your friends and brothers and all doing great in their life is something you always feel proud of. And today I really felt that Subhasis presented such a beautiful presentation, and I hope my students has been enriched with his knowledge. And for this, I am really thankful to Subhashis. I'm thankful to Sandipan sir to bring him to this platform, and I'm really thankful to all the students who were present today and who actually participated actively in today's session. Okay, and we also have our outgoing students with us, and it's really nice that this platform is there, and we are really getting into very good collaborative work with this kind of lecture series. Thank you, Subhasis. Thank you very much for being with us for this today's session. It was very humbling experience to have you.
Thank you, Ritesh da. Thank you, and I sincerely acknowledge whatever you have said. Thank you.